Good day, everyone, and welcome to our webinar on a new pilot program offering meeting opportunities to prospective generic drug applicants and generic drug applicants who intend to use model integrated evidence approaches for bioequivalence establishment in their abbreviated new drug applications. My name is Ray Ford, and I'm with the Small Business and Industry Assistance Program, also known as SBIA, which resides in CDER's Office of Communications, Division of Drug Information. I'll serve as your host for today's event. This slide provides QR codes for our social media avenues. You can scan them now with your phones and be taken to the appropriate platform. We'd like to share some logistics regarding today's 100% virtual event. There may be some internet issues, but please bear with us. If in fact we encounter those, we'll try to get them resolved as quickly as possible. Speaking of tech issues, the first slide provided the link for our one-stop shop for answers to your technical questions for downloading files and accessing this survey link to obtain the certificate of attendance and much more. That's the same page you used this afternoon for your online access to this webinar, so please bookmark that page. If you have any technical questions, please check there first and you'll most likely find the answer. We invite you to submit your questions to us prior to and during the productive Q&A segment, which is crucial to these events. We will answer as many questions as time allows, and when you do submit your question, please identify the individual presenter by preferencing your question with the speaker's name as shown here at Liang. There's a link on the right side of the pod where you see the speaker's headshot and it says download the presentation. Note that this link is only active during the real-time presentation. There's also a link to the speaker evaluation. We encourage you to take time to complete the evaluation as your feedback is much appreciated by the individual speakers. And in addition to this speaker evaluation, we have our final evaluation. Upon completion of this survey, an attendee can then download a certificate of attendance which may be used in support of RAP, SOCRA, SQA, and ACRP credits. The trick is that the certificate must be downloaded immediately upon completing the survey. Otherwise, this opportunity is lost. Even if you do not want or need a certificate, please take a few minutes to complete the final evaluation to help guide the design of our future events. Note that both the evaluation and certificate are only available for 2 weeks, which means that the deadline is February the 1st. The survey link will be posted on our one-stop shop page this afternoon. As a reminder, all the presentations are being recorded. It'll take us a week or two to process the recordings, but you'll soon be able to access them through SBI's homepage, and that's fda.gov/CDER Learn. To simplify things, SBI will send one email to Adobe attendees later today post-event. The email SBI sends will have information on the evaluation link. We'd like to take this opportunity to remind you of our upcoming events. On February the 13th through the 15th, we'll be providing a joint US FDA, MHRA UK Health Canada Good Clinical Practice and Pharmacovigilance Workshop, Compliance Workshop. February the 22nd, we'll have a webinar on the joint US FDA Health Canada ICH public meeting, and on March the 7th. Please join us for the Integrated Safety Analysis in Drug Marketing Applications, Avoiding Common Mistakes. Now we have a special announcement to make on enhancing the adoption of innovative clinical trial approaches to understand the state of innovation and clinical trial design and conduct. FDA is asking for perspectives via comments to our public docket, and that's FDA-2023-N-4489 by April the 19th, and or participation in the public workshop hosted in partnership with the Duke Margolis Center for Health Policy to occur on March the 19th and 20th. For more information, please reach out to the contacts listed on this slide. Now to start our presentations, it's our pleasure To welcome your presenters for this webinar, first we'll welcome for opening remarks Dr. Liang Zhao, Director, Division of Quantitative Methods and Modeling, Office of Research and Standards and Office of Generic Drugs, which leads us into our first presentation on MIE Pilot Program Process Overview by Maria Monroy-Osorio, Regulatory Health Project Manager, Office of Research and Standards and OGD. Next, we'll learn about potential topics for discussion through the MIE Industry Meeting Pilot Program by Dr. Andrew Babiskin, Lead Pharmacokineticist, Division of Quantitative Methods and Modeling, Office of Research and Standards and OGD, and the final presentation before our panel discussion is on considerations and expectations when meeting with the FDA under the Industry Meeting Pilot MIE program. This is by Dr. Eleftheria Saclazal, Senior Pharmacologist and Acting Team Lead, Division of Quantitative Methods and Modeling, Office of Research and Standards, and OGD. Please join me to welcome for opening remarks, Dr. Zhao.
Thank you, Ray, for the warm introduction. I'm Liang Zhao, the Director of the Division of Quantitative Method Modeling. In today's presentation, I will provide insights into the context, vision, key aspects, and anticipated benefits of the MIE industry meeting pilot. Quantitative method modeling stands at the cornerstone in the modernization of drug development, and its widespread application in generic drug development and approval encompasses in vitro and in vivo BE assessment, evaluation of drug device combinations, and post-market surveillance to gauge generic drug performance. The concept of model integrated evidence entails leveraging information generated by models, such as virtual BE study results. This approach goes beyond merely planning pivotal studies to play a pivotal role in providing critical evidence for drug approval decision-making. In OGD, the toolkit for modeling simulation methods encompasses PBPK models for both oral and non-oral drugs, conventional population-based PK, PK/PD, exposure response models, and big data AI machine learning tools for data analytics, risk-based models, and business process improvements. Before diving into the MIE pilot program, let's draw insight from our observations of the MIDD paired meeting pilot as experienced from the new drug side. Following its implementation in PDUFA 6 in 2017, we observed a gradual increase in the number of meeting requests over the initial 3 years, then plateaued in the 4th year. At the plateau level, a total of 16 meetings were requested, with 11 of them being addressed through written responses or face-to-face meetings. The industry has warmly embraced the program, as evidenced by the collaborative paper. The advantages encompass not only time and investment savings, but also improved alignment and enhanced clarity. It's crucial to emphasize that modeling simulation typically involve intricate methods, playing a premium on the necessity for alignment and clarity in order to navigate through their complexities successfully. In a broader context, when looking at the generic drug side, there has been a notable rise in the number of pre-ANDA interactions with a focus on modeling since the initiation of the PDUFA 2 period. This trend reached its peak in the 4th year of the PDUFA period, specifically between 2020 and 2022. The annual number of interactions has consistently ranged from 13 to 16. Would like to highlight the findings of a survey we conducted in October 2022. During this time, the FDA and the Center for Research on Complex Generics collaborated to host a well-attended live virtual workshop titled Utilizing Modeling Approaches to Support Generic Product Development. The workshop drew participants with highly diverse backgrounds, spanning drug product development, clinical studies, and regulatory affairs. Subsequent to the workshop, a survey was conducted to gather insight on the application for MIE for generic drug development. The survey primarily focused on gathering feedback on the workshop, and the overall responses were overwhelmingly positive and supportive of employing model-integrated evidence for generic drug development and approval. Notably, when asked about the value of establishing a communication channel with the FDA to discuss modeling approaches relevant to using the same model or modeling strategy across multiple submissions for complex drug products, no respondents answered negatively. The majority of responses expressed a positive affirmation for both the need for such a channel and its perceived value. After years of dedicated research, communication, and stakeholder engagement aimed at harnessing modeling simulation approaches to streamline and modernize generic drug development, we announced the official launch of the Model Integrated Evidence Industry Meeting Pilot on October 2023. In its mission, the new pilot program is to provide industry with meetings and opportunities for early interaction for science-driven topics using model-integrated evidence approaches for BE establishment to facilitate generic drug development and regulatory decision-making. In its vision, the pilot program allows enhanced scientific communications between generic drug developers and FDA on using a broad range of quantitative method modeling techniques to address generic drug development issues or questions that are either out of the scope or cannot be sufficiently addressed by the existing pre-ANDA and ANDA scientific meetings. You will find more details on the difference between pre-ANDA meeting and an MIE pilot meeting from Dr. Eleftheria Takozolozo's presentation. Specifically, the pilot MIE meetings will be more on discussing scientific and technical topics of using MIE strategies, while pre-ANDA meetings will be more on general scientific and regulatory issues. The MIE pilot will serve as a valuable communication channel with the FDA across three different scenarios. Firstly, it will address complex scientific or technical MIE issues relevant to either a single product or multiple products. For instance, discussions may revolve around common strategies for verifying and validating a computational fluid dynamics model or platform concerning the prediction of regional depletion of the inhaled aerosols. Secondly, the MIE pilot will encompass complex modeling approaches tailored for non-complex products, such as supporting BCS-based biowaivers and other BE study waivers that go beyond current recommendations. Thirdly, the pilot will extend to cover novel data analytics tools, including advancements in modeling methodologies or their applications. Examples include equivalence assessments of complex particle size distributions, novel quantitative approaches for API sameness assessment, and the utilization of cutting-edge data analytics methods such as machine learning for equivalence assessment. For further insight into these scenarios and the overall implementation of the MIE pilot, Dr. Andrew Babiskin will provide more detailed information in his upcoming presentation. The critical operational details of the meeting will be presented by Maria Monroy-Osorio following the pilot's launch on October 1st, 2023. The FDA will assess the program at the end of the first year or after conducting 5 meetings. whichever comes first to determine the subsequent phases of the pilot program. As highlighted earlier, the pilot will focus on both complex and non-complex products. Notably, we also anticipate the application of modeling strategies in the realm of oncology drug products, acknowledging the challenges in recruiting patients for steady-state crossover BE studies. Perceived benefit extend to both industry and the agency. For industry, these advantages encompass not only time and cost savings, but also heightened alignment and clarity, particularly when leveraging MIE approaches. This becomes particularly pronounced when a comprehensive understanding of a complex modeling approach cannot be fully achieved through pre-ANDA meeting alone. From the agency's perspective, adopting MIE approaches brings about operational efficiency in managing multiple products, reduces the number of review cycles, and fosters the development of an ecosystem with the industry to devise effective BE approaches. Of note, many of these benefits are mutually beneficial, creating a shared value proposition for both the regulatory agency and the firm involved. Following this introduction, our agenda will feature 3 presentations preceding the panel discussion and the Q&A session. Maria will delve into the process aspects. Dr. Andrew Babiskin will explore the potential subject or product areas that stand to benefit significantly from leveraging the MIE meeting pilot. Dr. Alvateria Sadalozo will further discuss the difference between a pre-ANDA meeting and an MIE pilot meeting, along with the insights on how to make the meeting productive and meet your need. Subsequently, a panel discussion will be facilitated involving key figures from the OGD and OPQ leadership. The discussions will revolve around the value propositions and the potential impact of the MIE pilot. Following the panel discussion, a Q&A session will provide an opportunity for the audience to pose technical, operational, and scientific questions. In closing, I want to extend my heartfelt appreciation to all the colleagues who have played a pivotal role in bringing the MIE industry meeting pilot to fruition and making it alive and accessible. This collaborative effort is truly a remarkable achievement for both the agency and the industry, and I'm confident that it will have a lasting and a positive impact. With that, I conclude my introductory remarks, and I'm pleased to pass the baton to Maria, who will delve into the intricacies of the process.
Good afternoon, everyone, and thank you for joining us for this webinar. I am Maria Monroy-Osorio, and I'm a Regulatory Health Project Manager within the Office of Research and Standards in the Office of Generic Drugs. Today, I will be discussing the process and anticipated timeline for the MIE Industry Meeting Pilot Program. During my presentation, we will cover the 3 stages of the MIE Pilot Program process. These stages include the meeting request and meeting package submission, in which the applicant requests a meeting with FDA, and FDA assesses the meeting request, concluding with a grant or denied decision. The second stage is the meeting preparation and conduct, during which FDA assesses the meeting package, determines the need for an optional orientation meeting, and conducts the final external meeting. And stage 3 is FDA's post-meeting communication, in which FDA communicates the final discussions held during the MIE external meeting. I will also touch on the expected timeline under the MIE pilot program. First, let's cover the 3 stages of the MIE pilot program. The MIE pilot program begins with stage 1. This stage consists of the MIE meeting request and package submission, which is approximately 14 calendar days. Stage 2 is approximately 120 to 150 days, depending on the request of an optional orientation meeting, and this stage covers the meeting preparation and conduct. And lastly, we will expand on stage 3, which is approximately 30 days and covers the post-meeting communication between the applicant and FDA. It is important to note that an MIE meeting request submission does not guarantee that a meeting will be granted. If a meeting is granted, at that point, stage 2 and stage 3 will occur. If a meeting is denied, the applicant may pursue other communication pathways that align with FDA's normal procedures. These communication methods can be controlled correspondences, as determined by FDA's guidance for industry, controlled correspondence related to generic drug development issued in December of 2022, or it can also be pre-ANDA product development meetings or pre-ANDA pre-submission meetings as described in FDA's guidance for industry, formal meetings between FDA and ANDA applicants of complex products under GDUFA issued in October of 2022. First, let's expand on stage 1. As I've said, stage 1 consists of the meeting request and package submission to FDA. Meeting requests should be submitted for complex or non-complex products and should include a focus on the proposed MIE bioequivalence approach, specific technical questions you wish to discuss with FDA, and include a complete meeting package as described in the following slides. Please note that at the time of submission, a full data set is not required but may be requested by FDA if the meeting is granted. Applicants should be prepared to submit the full data set to FDA within a reasonable time frame as requested by FDA. Lastly, we request that you not submit multiple meeting requests for an MIE pilot program, multiple controlled correspondences, or multiple pre-ANDA program-specific meetings for the same development product at or around the same time with the same or similar questions at which the first MIE pilot program meeting request is submitted. The Generic Principles Pilot Program has additional details on the type of meetings FDA will be looking for to grant under the MIE Pilot Program, and during the next few presentations, the speakers will cover key details on the expectations and considerations for MIE Pilot Program meetings, as well as the examples of the types of meetings FDA is looking to grant under this pilot program. When looking at the package contents, we first request a cover letter to the overall meeting package. In the cover letter, we ask that you indicate your interest in submitting an MIE pilot program meeting request. This cover letter should also include information on the pre-assigned ANDA number, the product name, the meeting objective, and applicant information. As we dive into the meeting package, please note that we have divided it into certain categories to help streamline your preparation of the package, the submission, and the overall FDA review of the package. The first category is General Information section. In this section, we request the pre-assigned ANDA number, the applicant information. If an applicant is outside of the United States, a U.S. agent must be appointed and a letter of authorization for the U.S. agent must be submitted along with the meeting package. Information and context on the product and development to further include the reference listed drug. And lastly, information on any previous interactions with FDA, such as controlled correspondences, pre-ANDA product development meetings, or pre-submission meetings. The next category is the meeting information section. In this section, we request that your proposed meeting agenda, your meeting participants, and identification of dates and times that are not suitable for a meeting with FDA. Please note that by default, all MIE pilot program meetings will be granted as video conferences. The last category of the MIE meeting package is the MIE utilization and approaches section. In this section, we request a brief statement on the purpose and objective of the request, answering the question why an MIE meeting would be beneficial, the specific questions for discussion about MIE approaches to establish bioequivalence, a statement of the questions of interest in the study or development program, A description of how the MIE bioequivalence approach will be used to address questions of interest and inform regulatory decision-making. Sufficient details for underlying assumptions and building process. And a clear model verification and validation strategy, including current and future data support. As I mentioned previously, a full data set is not required at the time of submission. However, it can be requested by FDA if a meeting is granted. If the request is made, the applicant should be prepared to provide the full data set to FDA within a reasonable time frame. I do want to highlight that the speakers after me will be discussing the meeting package contents in further detail, what FDA is looking for in the package, as well as determining why an MIE meeting would be beneficial to you. Once the meeting package is complete, you are ready to submit your meeting requests. Meeting requests and a complete meeting package is to be submitted to the FDA via email to mie@fda.hhs.gov with the subject line Request for MIE Pilot Program Meeting. Upon receipt of the meeting request and package, a confirmation of the meeting submission will be sent by FDA to the applicant via the same email address. In this email, the FDA receive date will also be confirmed. This date will be the business day or next business date when the meeting request and package was submitted and received by FDA. For example, if a meeting request and package is submitted on a Saturday, the FDA receive date will be the following Monday. The determination of the FDA receive date will also be taking into account any federal holidays. Once the meeting request and package are received, FDA will review the submission and provide a grant or deny decision. FDA strives that within approximately 14 calendar days of the FDA receive date, a decision will be made to grant or deny the meeting. The decision to grant or deny the meeting is at FDA's sole discretion, and a final decision will be communicated to the applicant via email. Please note that MIE meetings are not GDUFA-3 meetings and therefore are not subject to performance goals for scheduling and conducting meetings. FDA remains committed to the individual process and timeframes, and the MIE pilot program should not adversely impact FDA's ability to meet its individual performance expectations. If a meeting is granted, Stage 2 will begin. Stage 2 can consist of up to 2 video conference meetings between the applicant and FDA. The first meeting is an optional orientation meeting that will be scheduled approximately 30 days from the FDA grant date. This meeting is requested at FDA's discretion and is aimed at providing FDA with an overview of the proposed MIE approach and additional background and context related to the generic drug development process. The optional orientation meeting is a new element unique to the MIE pilot program and is not available in other meetings with FDA for generic drugs. This meeting will further help increase communication between the applicant and FDA and assist in guiding the meeting package review and overall final external meeting conduct. If an optional orientation meeting is requested, the applicant will be notified via email, and the meeting will be scheduled within approximately 30 days from the FDA grant date. Formal meeting minutes for the optional orientation meeting will not be issued. If an optional orientation meeting is held, the timeline of Stage 2 may be extended if FDA, at its sole discretion, deems that additional time is necessary for a meeting package assessment and preparation for the final external meeting. This additional time will be communicated via email to the applicant. The next component of Stage 2 is the preliminary responses. The preliminary responses will be issued to the applicant via email approximately 5 calendar days prior to the final external meeting. The preliminary responses are aimed at addressing the applicant's questions prior to the final external meeting. Once received by the FDA, the applicant has 2 options to proceed with. The first option is to notify FDA that the preliminary responses provide a meaningful written response to the meeting package and a video conference meeting with FDA is not needed. If an applicant wishes to cancel the final external meeting and accept the preliminary responses as the final official record of the MIE pilot program meeting, they can provide a request for cancellation via email. In this email, we request that you indicate your decision to cancel the final external meeting and accept FDA's preliminary responses as the final meeting response. The second option is to provide an updated agenda to the final external meeting if discussion is needed for only some of the original questions. If the applicant wishes to pursue this pathway, please send the updated agenda with questions for discussion and clarification via email no later than 48 hours prior to the final external meeting. The updated agenda and clarifying questions will be discussed during the final external meeting. The final component of Stage 2 is the final external meeting. This meeting will be scheduled within approximately 120 days from the FDA grant date. If an optional orientation meeting was held and FDA determined additional time was needed for review of the meeting package, the final external meeting will be held within approximately 150 days of the FDA grant date. If this is the case, the applicant will be notified at approximately day 60 that an extension was made. The final external meeting will follow the updated agenda and clarifying questions as determined by the applicant after receipt of the preliminary responses and will be held in accordance with FDA formal meetings. As a reminder, the scheduling of the meeting and timeline is approximate and not subject to performance goals under GDUFA 3. However, FDA does remain committed to the individual timeframe for the MIE pilot program. Stage 3 is the post-meeting FDA communication. This stage covers the post-meeting communication between the applicant and FDA. The applicant may submit a meeting summary of the final external meeting within 7 calendar days of the meeting date via email, and FDA strives to issue the final meeting minutes within 30 calendar days of the meeting date. The FDA meeting minutes are the official record of the final external meeting date. In summary, the MIE pilot program consists of 3 stages and the overall timeline is dependent on the granting of the optional orientation meeting. Stage 1 covers the meeting request and package submission and a grant-denied decision and is approximately 14 days from the FDA receive date. Stage 2 covers the meeting preparation and conduct and can consist of up to 2 video conference meetings. If an optional orientation meeting is not granted, the final external meeting will be scheduled within approximately 120 days of the grant date. If an optional orientation meeting is granted, then the final external meeting will be scheduled within approximately 150 days of the FDA grant date. Regardless of the scheduling of the final external meeting, the FDA preliminary responses will be issued approximately 5 calendar days prior to the final external meeting. Stage 3 covers the post-meeting communication between the applicant and FDA. The applicant may submit their meeting summary within 7 calendar days of the final external meeting, and FDA strives to issue the final meeting minutes within approximately 30 days of the final external meeting. As a reminder, the MIE meetings are not GDUFA 3 meetings and therefore are not subject to performance goals for scheduling and conducting GDUFA 3 meetings. FDA, however, does remain committed to the individual process and timeline as indicated on this slide. I want to thank you all once again for attending today's webinar. I will be back on the live Q&A session at the end of today to discuss any questions you may have. As always, if you have any questions, please email mie@fda.hhs.gov.
Everyone, my name is Andrew Babiskin. Today I'll be presenting on potential topics for discussion through the Model Integrated Evidence, or MIE, Industry Meeting Pilot Program. While the pilot program materials on the website give some examples, the goal of this presentation is to provide you, the audience, with a more thorough list of MIE-based approaches to educate you not only on what type of topics/issues can be brought into these industry meetings, but also on the type of topics/issues where MIE can be applied for generic drug development. Before we get into the rest of the presentation, this slide is to remind you that this presentation reflects the view of the author and should not be construed to represent FDA's views or policies. You've already seen this slide in the earlier introduction presentation from Dr. Liang Zhao, but with slight modification. The three major MIE areas that could be applied to generic drug development are depicted here. These three areas being, first, mechanistic modeling such as physiologically-based pharmacokinetic modeling or PBK modeling, and computational fluid dynamics that can be applied to both oral and non-oral drug products. Next is pharmacometrics and quantitative clinical pharmacology, which includes population-based modeling and pharmacodynamic modeling. And lastly is data analytics, which was referenced as big data in Dr. Zhao's earlier presentation, but these include machine learning and analytics for complex mixtures, for example. You will see that as I continue into the potential topics portion of this presentation, that the topics are organized in these same categories, with mechanistic modeling split out separately in the oral and non-oral areas. I want to provide you quickly with the goals of the MIE Industry Meeting Pilot Program. The text on this slide is taken directly from the General Principles document provided on the FDA website and whose link is provided on the slide below. I recommend you read that document, but also other presentations and discussion today will focus on this content. Namely, I want to emphasize that the main goal of these meetings is to enhance scientific communication in the areas where MIE is being applied to generic drug development. These approaches are largely novel and innovative, and would benefit from the increased interaction with the quantitative discipline experts. The General Principles document also contains a list of the situations in which an MIE industry meeting may be granted, which is again provided here verbatim, along with a link to the General Principles document for your reference. These situations include, first, innovative MIE-focused approaches for bioequivalence establishment that cannot be effectively addressed under the existing GDUFA scientific meetings. These innovative approaches can be applied across the full range of products, including both complex and non-complex generic drugs. In the presentation that follows mine from Dr. Eleutheria Zakalozu, you will hear more about the differences in utilizing the GDUFA pre-ANDA product development meeting versus this MIE pilot program industry meeting. The next situation, which is non-complex products with complex approaches / modeling for biopharmaceutics classification system or BCS-based bio-waivers and or other study waivers. This situation is more specific and deals with the application of study waivers for non-complex products such as oral drug products. You will hear more about this later on in the potential topics for the use of PBK for oral drug products. The last situation is novel data analytic tools and approaches, for example, machine learning and artificial intelligence for bioequivalence establishment and assessment. With these situations in mind, the 4 quantitative disciplines in the Division of Quantitative Methods of Modeling curated a list of potential topics for the pilot program in each of these areas. These will be presented in the order of oral PBK, non-oral PBK, quantitative clinical pharmacology or QCP, and data analytics. The list of topics presented here today is not meant to be an exhaustive list, but is one developed through various types of previous industry interactions, such as pre-ANDA meetings and control correspondences, that we found would benefit from the MIE Industry Meeting Pilot Program and were really the source of inspiration for developing this pilot in the first place. Before we enter into the list of topics, if you have specific questions for the potential topics in the different discipline areas, you can address those questions to me to identify this presentation. But I want to note that in the Q&A to close out this webinar, I will be joined by other modeling discipline leads in DQMM who will assist me in addressing all your questions. The first topic area for today, we will go through the use of PBK for oral drug products. The area of oral drug products, PBK modeling-based approaches can play various roles in your application. PBK modeling can be used as a risk assessment tool for justifying the impact of deviations in vitro dissolution profiles or particle size distribution on bioequivalence. For alcohol dose stuffing studies, PBK modeling can evaluate the impact in dissimilar dissolution in the presence of alcohol. PBK absorption modeling can also be a critical tool set in the establishment / identification of biopredictive dissolution methods to support bioequivalence evaluation. A big topic these days with the publication of the M13A ICH guidance is the waiver of fed bioequivalence studies for oral products. PBK modeling can be used to evaluate the impact of food for products that have been defined as high risk. PBK modeling can be utilized to support biowaivers for bioequivalent studies in subjects with gastric pH change. For example, when the B study is to be conducted in the presence of a proton pump inhibitor, when such a study may be necessary for establishing bioequivalence. PBK modeling can also be used to support BCS-based biowaivers, for example, in evaluating the impact of excipients on bioequivalence for BCS Class 3 products. Lastly, PBK modeling can be used to justify your study design, such as when only single-sex data is available for B evaluation, or evaluating the sensitivity of the analyte to use for bioequivalence. We next move on to the topic related to mechanistic modeling for non-oral products. Potential meetings could discuss for orally inhaled drug products, model validation of regional deposition models, and the relationship of in vitro measures to drug at the site of action. For topical dermatological products, topics could focus on the validation of dermal PBK models and in silico in vitro permeation tests or IVPT models using local and systemic drug concentration data. Meetings can also discuss platform validation for a specific set of locally acting products, such as topical dermatological products, which would show the performance of the platform to predict a multitude of different drug products. For in silico IVPT models for topical dermatological drug products, topics can include the development and validation of these models for products where in vitro data generation is challenging, and also the application of these models to inform in vivo dermal models through in vitro and vivo extrapolations. For non-oral products where drug release is driven by unique formulation characteristics such as PGLA-based long-acting injectables and ophthalmic inserts, the development of validation of release models can be a potential topic for discussion within the pilot meetings. There has also been interest in using non-clinical data to support model development and the bioequivalence determination for certain products such as ophthalmic and LAI drug products. The MIE meetings can be used to discuss validation and application of interspecies model extrapolations with PBK modeling. Lastly, development of PBK models for LAI products can be discussed, which account for the interplay of formulation attributes and local physiology on in vivo release. The next area relates to use of quantitative clinical pharmacology and virtual bioequivalence in that context. One major application area of QCP/population PK modeling is for development of alternative study designs with either shortened overall duration or decreased number of subjects or even both. Examples of products that may benefit from shortened overall duration are LAIs and for studies carried out in steady state in sensitive patient populations. MIE pilot meetings can be utilized to discuss a number of different POP-PK-based approaches to justify a shorter study duration, such as in silico continuation, where steady state is simulated from a study where steady state is not achieved, carryover adjustment when there isn't sufficient washout between subsequent doses, and truncated study design with narrow B limits. These are just a couple examples, and others are listed on the slide. Examples of products that may benefit from having to recruit and complete less subjects are orphan drugs, and again for those products where studies will be conducted in sensitive patient populations. Meetings can discuss innovative ways of applying reference scaling and also sparse or reduced sampling optimization. There are some practical study considerations and bioequivalent assessment challenges that can be addressed through population PK modeling. The application of such models to solve these challenges can be a topic of discussion in the MIE pilot meetings. Examples of these challenges include verifying steady state attainment, data imputation to address missing samples in either PK or PD studies, model-based adaptive design, justification of deviations in PK profiles and the impact on bioequivalence, and determination of Emax for dose scale PD studies. Lastly, another topic for discussion is the application of PK or bioequivalence bridging to an alternative product when a reference standard does not exist due to discontinuation. Final area for today is the use of data analytics and machine learning. Potential topics for discussion in the pilot include complex comparative profile analysis, which for example would include analysis of complex particle size distributions in LC-MS profiles. Second is complex substance analysis, which can include novel quantitative tools for analyzing the active ingredient. And lastly, third is novel data analytic tools that incorporate artificial intelligence, for example, machine learning, to support data analysis and/or model building in the eventual ANDA submission. In the previous slides, the topics were largely focused on the applications at MIE, with some mention of some specific modeling steps such as model or platform verification that could be explored through the MIE industry meeting. Regardless of the modeling discipline, your topics can focus on one or more modeling steps. These steps include, first, model or platform development. Discussion can center around justification of model input parameters like how a formulation is defined, for example. Second step, as already mentioned, is model or platform validation, where discussion should be on the appropriate level of validation in the model with the intended regulatory use in mind. And lastly, the model application. This could be discussions on the intended virtual bioequivalence simulations and the appropriate plan and/or design of those trials. The subsequent presentation from Dr. Zakaluzu will delve more into the meeting package expectations with regard to these elements. For my presentation today, there are several take-home points that I summarize here for your use. First, I would like to reiterate that the pilot program includes both complex and non-complex products, and that the discussions that take place in these meetings may pertain to a single product or even a class of products. In the 4 modeling disciplines presented here today, please refer to those examples as guidance on what issues this pilot program serve as a benefit to your generic development program. Again, all these topics here are not meant to be exhaustive, and we expect that generic industry may have further uses of MIE to discuss. Thank you for listening to my presentation, and I look forward to answering your questions alongside my colleagues in the Q&A.
Hello everyone, my name is Eleftheria Tsakalozou. I'm a senior pharmacologist at the Division of Quantitative Methods and Modeling, the Office of Research and Standards, the Office of Generic Drugs at the FDA. After a very informative presentation by Dr. Babiskin on the potential topics that can be handled under the Meeting Pilot MIE program, we can now move on to discuss considerations and expectations when meeting with the FDA under the Industry Meeting Pilot MIE program. My disclaimer: this presentation reflects the views of the author and should not be construed to represent FDA's views or policies. My talk is divided into 2 parts. First, I will go over the components of an effective FDA industry meeting, what is the meeting process, the different stages, and how the applicant can maximize the impact of the Model Integrated Evidence MIE pilot program. The second portion of my presentation will focus on the differences between the pre-NDA product development meeting under GDUFA and the current MIE pilot program meeting. This portion of the presentation will help you decide which one is right for you. Very briefly, the goal of the MIE Industry Meeting Pilot Program is to enhance scientific communications between generic drug developers and the FDA on using a broad range of quantitative methods and modeling techniques to address generic drug development issues or questions. Through this program, applicants can obtain the FDA's advice on how their proposed modeling approach can be used in a specific drug development program, how they can advance modeling methodologies to address common issues of multiple products from the same applicant, and how to address complex issues as they arise and implement innovative approaches in the development of non-complex products. The key points that I will be discussing today and are captured in my presentation are offered by scientists in the Division of Quantitative Methods and Modeling, scientists that have led or supported several FDA industry interactions that were focused on modeling and simulation approaches. These interactions mostly took place under GDUFA 1, 2, and 3, and they include pre-NDA product development and pre-submission meetings, mid-cycle review meetings, post-CRL scientific clarification meetings, product-specific guidance teleconference meetings, as well as other interactions such as control correspondences and ANDA assessments. Let's move on now to the first section of my presentation and discuss what are the components of an effective meeting between the FDA and industry. What are the different meeting phases and how to maximize the impact of the MIE pilot program meeting. The FDA industry meeting process can be divided into 3 phases. The pre-meeting phase is initiated by an applicant that submits a comprehensive meeting package. The agency may follow up with an information request. Prior to the meeting, preliminary responses are shared. These preliminary responses should be concise and informative. The meeting is held at an agreed and reasonable time frame, allowing for FDA assessment. The second phase of this process is the meeting phase. All parties join the meeting with the intention to share all relevant information. Open discussion takes place during the meeting within the scope of the meeting objective. And by the end of the meeting, parties have achieved clarity on the critical aspects of the discussion and are satisfied with the responses they received. Finally, the meeting outcomes are documented in the form of meeting minutes. Diving a little bit deeper now into what makes an MIE pilot program meeting successful. Let's discuss what should be the content of the meeting package that will be submitted, who should be invited to the meeting, and when should a meeting request be submitted. Let's begin our discussion with the meeting package preparation. The meeting package should contain a sufficiently developed scientific proposal. The meeting question should be relevant, critical, and clearly stated. The modeling approach should be adequately documented in the meeting package. The model objective should be clearly stated. Assumptions and justifications relevant to model building should be included, as well as a risk analysis assessment when applicable. In certain cases, case studies that demonstrate the modeling approach may be extremely helpful. The meeting package should contain adequate documentation of the modeling approach for each proposed regulatory use. A very important component of the documentation is model validation, which should be appropriate for the proposed modeling approach. For instance, model validation often focuses on only one drug product, but validation is needed for multiple products to demonstrate model sensitivity to product differences. In other cases, model validation may occur using a different drug product or product type, DPIs versus MDIs, for instance. The model application scope should be clearly defined and described for the drug product of interest and future model uses, if applicable. Finally, when the modeling package includes an approach for virtual bioequivalence analysis, components of the virtual bioequivalence analysis, such as the selection of subjects, the power of the study, different types of variability should be included into the meeting package, as well as a risk assessment that may include type 1 and type 2 error analysis. It is important that the meeting package is error-free and it contains sufficient information to replicate the analysis that it describes. That will limit the need for information request, as well as the need for orientation meetings that extend the assessment time. Significant components of the meeting package are the model analysis plan and the modeling report that support its regulatory use. Model verification and validation criteria should be pre-specified. The model building and development criteria, including formulation effects, should be pre-specified in the meeting package. When model files are submitted, they should be provided in an organized fashion for accessibility. When datasets are submitted, summary or individual level data may be submitted as appropriate. Supporting data may also need to be submitted, such as study synopsis and reports. Moving on now to the second phase of the process, the actual meeting. An open discussion within a multidisciplinary team from the FDA and industry will be taking place during this phase. Attendees should be knowledgeable of the technical details related to the proposed modeling approach, familiar with the drug product or products of interest, and familiar with the regulatory context. Another important question is when to submit a meeting request. The agency encourages applicants to engage early in their drug development program. This is feasible under the MIE program because the burden for preliminary data accompanying the package is low. Also, it is important to have a clear understanding of the role of the MIE approach on the overall regulatory program early. A scientific discussion can take place on novel model-based B approaches under the MIE program, and this can benefit the drug development program. However, the meeting package that will be submitted should be complete. The early engagement is beneficial also because meeting outcomes can be generalizable across more than one product in the pipeline. Now I would like to move on to the second part of my presentation and discuss potential differences between the pre-NDA product development meeting and the MIE pilot program meeting. Hopefully this portion of my presentation will help you decide which one is right for you. There are differences in the scope between the pre-ANDA product development meeting and the MIE pilot program meeting. More specifically, modeling approaches for non-complex products with complex approaches are within scope for the MIE pilot program meeting. Model engaged biowaiver approaches for a certain type of products or study are also within scope for the MIE pilot program meeting. Innovative MIE-focused approaches for bioequivalence establishment to address complex issues for a single product or multiple products can be discussed under the MIE pilot program meeting. Novel data analytic tools such as modeling methodology advancement or new applications of the modeling approach, new quantitative approaches for comparative analysis, and application of novel data analytic approaches for equivalence assessment are within scope for the MIE pilot program meeting. Opportunities and challenges associated with the model master file can be discussed under the MIE pilot program meeting, as well as in vitro experimental methodologies when these are utilized for model development and validation processes. Here I would like to share some samples of potential deficiency languages that are within scope for the MIE pilot program. We recommend you use a biopredictive dissolution method to mimic fasted condition and use appropriate approaches to incorporate dissolution data into your PBPK model. You did not challenge the model with in vitro and in vivo data which showed lack of bioequivalence and or batches with different release rates to support the robustness of the established PBPK model. You should evaluate different clinical study designs and determine which ones will be the most sensitive and efficient for the population PK model to detect the formulation differences that would not lead to biased equivalence determination for the subsequent bioequivalence study simulation. You should indicate how the proposed MIE approach can properly characterize the uncertainty and propose the most appropriate BE statistical method. You should pre-specify the model analysis plan. Sufficient model verification and validation should be included for the intended regulatory purpose. Within the scope of the platform validation process, the drug product selected should involve active pharmaceutical ingredients with a range of physicochemical properties containing the API of interest. And finally, your proposed virtual bioequivalence trial lacks information on the study design, the virtual population, the sample size, the incorporation of inter and intra-subject variability as applicable, the statistical analysis you intend to perform for BE testing. Regarding the meeting process, an important difference between the pre-NDA product development meeting and the MIE pilot program meeting is the orientation meetings. These are readily available under the MIE pilot program meeting, but they're not common under the pre-NDA product development meeting. When it comes to the meeting time, we do encourage early engagement with the agency, and especially under the MIE pilot program meeting, this is feasible because the burden for generating preliminary data in the package could be lower in the MIE pilot program meeting when compared to the pre-NDA product development meeting. A complete alternative bioequivalence approach on the comparison between the reference listed drug and the test product is not necessary under the MIE pilot program. And the package can contain modeling considerations that are focusing primarily on the RLD in the case of the MIE pilot program. However, this is not the case for the pre-NDA product development meeting program. I would like to conclude this presentation by offering some take-home messages. To maximize outcomes from the MIE pilot program, sufficiently develop and adequately document your scientific proposal. Work with a multidisciplinary team. Engage with the FDA early in your product development program. Understand how the MIE pilot meeting program may be a suitable choice for your needs. A closing thought. Please consider the industry meeting pilot MIE program when requesting a meeting with the FDA. I'll be very happy to answer any questions during the panel discussion. For now, I would like to thank you for your attention.
Thank you for those great presentations. You may have noticed a slight shift in your screen for your computer. We are accommodating videos for all of our panelists and the moderator as we give our panelists some time to connect their videos and mics. We'd like to welcome our panel moderator, panelists, in addition to our presenters that'll be a part of the panel discussion. Our moderator for the panel discussion will be Dr. Lucy Thong, Deputy Director, Division of Quantitative Methods and Modeling, Office of Research and Standards in the Office of Generic Drugs. And she's already on screen there. As far as our panelists, we're welcoming Dr. Bhagwant Reddy, Division Director, Division of Biopharmaceutics, Office of New Drug Products in the Office of Pharmaceutical Quality. Also would like to welcome Dr. Partha Roy, Director, Office of Bioequivalence in the Office of Generic Drugs. And also to our panel, we'd like to welcome Dr. Robert Lamberger, Director, Office of Research and Standards in the Office of Generic Drugs. I'd like to take this opportunity to turn it over to our panel moderator, Dr. Fong.
Hello, good afternoon. My name is Lucy Fang, and it's my great honor to moderate this panel session. First, let's welcome our panel members. For this panel session, we will start from some panel questions based on my discussions with internal and external colleagues. And I personally very much look forward to hear the comments and inputs from this group of leaders on how MIE pilot program can enhance the generic drug development and best practice. After that, if time allows, we will try to entertain some audience questions. So I strongly encourage you to put your questions in the chat box. Even if we don't get able to address your question today, we will try our best to address those questions in a different venue. With that, let's come to our first question, which is shown on the screen right now. Can you share any past experience or examples that highlight the positive outcome of the use of MIE approaches in generic drug development? So, Dr. Zhao, we understand that Division of Quantitative Methods and Modeling is the expert in this domain and also initiated this pilot program. So can we start from you?
Yeah, sure. Thank you, Lucy, for giving me the chance to address this important question. We have been constantly improving the process communication between industry and FDA to facilitate generic drug development approval, especially when we are facing innovative approaches like modeling simulation. However, every innovation poses some challenges, especially during the initial phases. Modeling simulation, like model integrated evidence approaches, are inherently complex. They require time and effective communication to explain their role in the regulatory pathway, their technical nuances, their validation and verification strategies. We have learned this need from both new and generic drug sides. For instance, similar needs in new drug development led to the introduction of a new meeting type under PDUFA 6, the MIDD paired meeting pilot program. In the realm of generic drugs, experiences from pre- and post-CRL scientific meeting programs have shown that often over an hour is needed to adequately communicate about the modeling component. The IR mechanism alone cannot meet this need. After gathering feedback from industry stakeholders, we initiated this pilot program as a mechanism to address all related strategic or technical questions for generic drug developers. The goal is to reduce both the number of cycles leading up to final drug approval and post-submission meetings. A testament to the positive outcome is evident in cases like long-acting injectables, where agreement on regulatory pathways and the technical details can be agreed upon at the early stage. I think that will save up lots of later stage communication and surprises. With that, I want to conclude my comment. Thank you again, Dr. Fang, for the opportunity.
Anyone else in the panel also want to comment and share some experience? Okay, if not, we will come to the second question. We probably can come back to this question later. Okay, so the second question is, from a strategic standpoint, when do you see the MIE approaches making a significant impact on generic drug development and regulatory assessment? For example, like under what instances and what product categories might benefit more from this approach? So, Dr. Reg, I probably will direct this question to you first. We all understand that your division does a lot of in vitro, in vivo correlation and biopharmaceutics modeling. So really want to hear your thoughts first.
Sure. I think this was mentioned previously by the speakers in their presentations, but I'll just give my personal opinion. In terms of product categories, I think it's applicable to both complex and non-complex products. Specifically with respect to complex products where you cannot really measure the drug at the site of action. Those are the locally acting products, inhalation, dermal products. And those typically require clinical endpoint studies to demonstrate equivalence. That can be really large studies and not as sensitive. So I think this modeling and simulations approach will definitely be useful there. Similarly, long-acting injectables or even like intrauterine devices, like contraceptive devices that have 2 years of efficacy. It's difficult to conduct bioequivalence studies over a long period of time. So I think model integrated evidence will be very useful in those cases. Non-complex products were also mentioned, oral PBPK models in certain situations like BCS biowaivers, or if a reference product is not available. Those are the cases where they could be useful. In terms of when you should consider modeling approaches, hopefully you would want to develop regulatory strategies around it and submit those requests as early as you can before you submit your ANDAs. I think what we see, the issues that we see are in model development and validation. And a lot of times when the submissions come to us with models already applied, it's generally too late. So if you can get your feedback on what are the appropriate model inputs and what are appropriate studies to validate your models before you go deep into application of the models, I think that will be certainly useful. It could also potentially be useful in post-approval space. There are post-approval changes that are made routinely. New sites are added. Sometimes processes are changed for more efficiency, or newer advanced manufacturing technologies are employed. And in some cases, when those changes are to the extent of level 3 changes as per SUPAC, generally BE studies are needed. But again, if you incorporate modeling and simulation strategy in your development program and come in with either IVIVC proposal or virtual BE trial proposal based on PBPK modeling, before you implement those changes, that potentially could be very useful for you. So those are some of my thoughts on that questions. Thank you.
Thank you so much, Dr. Rich. So, Dr. Roy, your office, that's the ANDA reviews and really has seen a big picture. So I'm wondering, do you have anything to add?
Yeah, so thank you very much, Lucy. And I think Bhagwan probably has already covered many of the product categories that we have seen. I think I wanted to give you a little bit of a perspective also. I'm representing the Office of Bioequivalence and the entire assessment teams for bioequivalence. And then obviously we work very, very closely with our colleagues in the DQMM and ORS in general, and it's a more like a collaborative assessment approach. And these are the new approaches that are increasingly we are seeing a lot more in our submissions. So one of the I think in with this MIE pilot, what we are trying to do is also bridge two things. One, we are probably seeing a lot more instances of requests for modeling and simulation approaches in different product categories coming in the pre-IND stage. But then when it goes to the ANDA stage, sometimes we see that maybe because of the lack of information or the information that might be requested, and the companies may go the other route and just maybe using just the PSG approach that is already in the PSG, but not undertaking some of the novel approaches. So I think this is a meeting where you can actually have the discussion, in details, talk about the data requirements and what is actually needed. And it's important that also the bioequivalence reviewers, the entire review team, is present in some of these meetings early on. So I think that's where we see that a lot of the requests coming up front, and there is obviously a great interest in that. But then what we are trying to do is translate into real, within the ANDA side, actually make it happen and use this more and more to bridge the information. The other point I wanted to also focus on, because a lot of the times, I hear that, oh, if I use this, then can I just, replace a certain trial? And a lot of the times it's the clinical endpoint trial, which is obviously a big goal. And I understand that. But oftentimes I think it's important to kind of understand, from a regulatory approach, what is your regulatory strategy and how we are taking a stepwise approach to bridge some information which may be lacking as you go through your development journey. I think, and there are many, many cases, I think you heard from Andrew, and also you heard other presenters talking about different areas where modeling can be extremely helpful to bridge some of the information that is needed, and in particularly in some of those topical products. We have a particularly inhalation product, for example, we have a weight of evidence approach. So a lot of the time the modeling and simulation really can help inform us and actually reduce the uncertainty and the risk in the development, and that's where you can actually utilize that. And this is a meeting where you can have that discussion, very openly, and understand what are the limitations, and actually you can discuss, because we understand that early in the program you may not have all the data, and you are probably trying to figure out what else do you need to do to go over the hump, rather than doing an expensive, resource intensive clinical endpoint trial, for example. I mean, those are the discussions that you can have early on. And I think this is the pilot where we allowing this in a pre-NDA stage. But I think Bhagwat already mentioned that there is also probably an importance of it in the future, which we can bring it in if it is successful, even in the post-submission stage or after a complete response letter, for example. So those are the sort of the initial thoughts. Maybe I kind of deviated from the questions itself, but I just wanted to kind of lay the land here. Thank you.
No, this is excellent. I really appreciate a lot of your inputs. So, Dr. Zhao, I have seen you are nodding your head from time to time. Do you have anything else to add?
I don't have. I think Partha illustrated very well, especially there are big goals and also relatively lighter goals when using modeling simulation. Model integrated evidence approach can fit into many purposes. It can be targeting the weight of evidence approach to replace to conduct in lieu of comparative clinical endpoint study, for example, especially for already inhaled drug product. On the other hand, it can meet certain smaller purposes like justify for a dissolution profile deviation or justify for a particle size distribution difference from the formulation. I think modeling simulation is going to meet all those needs because it's not only a modeling, but more like a summary of knowledge and data from all angles. Then if FDA got enough data to support, to validate the model, then certainly that can abbreviate lots of programs. I'll stop here and give, probably we haven't heard from Dr. Rob Lamberger yet. I believe he has much more deeper insight into that.
Okay, thanks, Liang. Okay. So from a strategic point of view, when is it really useful to do this? And I can think of several cases on a broad level. One is when you're developing an alternative bioequivalence approach. So, our product-specific guidances are very important. If you're going to do the studies that are in the product-specific guidance, then the approach is pretty clear. But certainly we're open, through various ways to discuss alternatives to our guidances. But I think using a model approach is really important in that case, because how else do you justify that your alternative approach is valid, right? You could do a whole bunch of studies, but I mean, this is the generic program. It's an abbreviated pathway. So, and I think this is also something that speaks to our internal experience, is that many, many times when we are developing PSGs that are novel or new, we use the modeling approach to help us understand what the limits should be, how sensitive the approach is, how well it describes the data that's publicly available in the literature, and that. So we internally have found that incredibly valuable. But what that also means is that if you send us an alternative approach and we're going to look at it, we're likely internally going to say, well, let's use our modeling tools to evaluate this internal approach to see if it actually makes sense, because we think that's the best way to do that. And by participating in that, doing your modeling, you actually have the opportunity to, understand maybe what we're going to be concerned about and address that. So I think that anytime, so from a strategic point of view, if you're taking a strategy, I think I want to pursue an opportunity for an alternative to our PSG for either a complex or non-complex product. A model-based approach can be extremely valuable in supporting that. And using this meeting means you get some time to sort of align with what the best model for this case is, and you get the input from the FDA experts there. And then that really can make it easier to have the discussion around, all right, is my alternative approach okay? Because I've sort of set up the modeling things and had the modeling discussion through this new pilot program, then that discussion becomes clearer and more precise. If you just send in a model without without using this process, the first thing FDA reviewers do is say, well, wait a second, I better check that model. And so am I spending all of my meeting preparation time checking the model relative to actually getting to your regulatory decision point? So I think this meeting can be extremely valuable anytime you're thinking of proposing an alternative approach. Have a meeting on the model-based part that you're going to use to support that alternative approach. I think that can be a really valuable strategic direction. Another one, it was touched on a little bit by some of our other speakers, is, if you look at our PSGs, a huge trend over the last 10 years of GDUFA and the GDUFA regulatory science is making available Q3-based approaches for complex products. I think we've made huge strides in making these pathways open. We've approved lots of products using them. But one thing we've also no approach is perfect, right? Everything has something, every new thing you do opens up new challenges. And so one of the things we've seen across the Q3 space is, as you begin to measure more things, suddenly you say, well, look, I see a little bit of difference in the particle size. So our first Q3 approaches that have approved the products, they said, well, compared to the RLD, look at multiple batches of the RLD. If you're within them, then you're okay. Right? But not every case fits in that situation. Some RLDs are very precisely controlled. And you might say, well, look, I'm a little bit different than the RLD. Every RLD batch is very, very well controlled. What's my justification for this difference? We have some baselines for in vitro approaches of 10% differences are generally assumed to be, minimal given how we control most in vitro things. But beyond that, if you see some difference and you want to say, well, how important is this difference? A modeling and simulation approach can be extremely valuable in saying, no, in order for the particle size to change the local availability, you've got to double the particle size. Don't give me trouble because I have a 9% difference, right? Or that. But as part of the strategic approach to develop these, if you're heavily involved in the Q3 characterization approach for complex products, having the ability to have this modeling and simulation infrastructure set up on those critical attributes to understand how they predict the things that you can't measure directly, the local availability, can be really important and critical. I'm not saying this is easy because, like I said, we're targeting the local availability. It's really hard to measure directly. But we've invested through the research program significant amount of effort in developing tools for local delivery of various routes that are important to us: the inhalation, nasal, topical, ophthalmic products, and building out this model infrastructure, making that available. It's available in commercial software with tools to use. Just because it's available in commercial software doesn't mean it's going to be absolutely correct or acceptable. But that's what this meeting is for, right? I'm building up the infrastructure for my topical product development programs, and I want to understand how much, I want to have some confidence in being able to set limits on the globule size distribution that I measure. And my modeling says, look, it doesn't have to be precise because I need large differences to have an effect on local availability. That's the kind of discussion that can be very valuable to you as you get into situations where perhaps you're outside of like, well, look, I measured the RLD and I'm a little bit outside that RLD limit. How do you evaluate that? The modeling approach can be really key to helping make that decision and moving through that. But the modeling only helps you when FDA believes that the model is appropriate for use. And this meeting is really the valuable place where you can set that foundation point. Now, sometimes maybe you realize you're in the situation late. You can still use this meeting and say, well, look, here I'm in this situation. I really want to have the model in place. You can have, we're open to, as we're piloting this, learning what works best. you're open to having this meeting later and say, well, look, I have a difficult review situation here. I want to use this model. Let's have this model meeting first, right? Make sure the model is acceptable. Then you can have the discussion about solving the regulatory problem. So I think those are two really important strategic parts of your development program that you can use this meeting for to enable those types of supporting things.
Thank you, Dr. Leinberger. Actually, we have a visionary question for number 3. We were thinking that you probably will help us provide some input. I think you kind of touched upon some of the aspects when you were trying to address question number 2. So the question number 3, I will be quick, is what specific benefits do you foresee this MIE pilot program bring to the generic drug program? And really want to touch upon, are there any particular challenges that you think this pilot meeting program can address? So really trying to help our industry colleagues to understand the unique benefit for this new initiative.
Yeah, I mean, I think the practical part of this meeting, apart from the strategic direction, the practical part is what we've noticed is when you send in a complex product, when you send a complex model, FDA staff spend a lot of time trying to understand, well, what did you do? What data did you do? What's the right validation standard or verification standard for this model? And this can be a limiting factor, right? In a normal pre-ANDA meeting, if we have 120 days with your package and 90 days are spent on the model, maybe we miss the time that we need to discuss your regulatory question. Or in the context of an application, right? If you send us this complex model in the application, then somebody's sending a consult, we're developing who's going to evaluate the model. You have to evaluate the model. Then you have to loop back with the review team, say, well, all right, now we've answered our questions about the model. We actually believe that it's appropriate for this use. We've gotten more information on that. Then you're back to what's the review question, and you can be up against timelines there. So I think this very specific benefit here is having the ability to separately address questions of, the model's fit for purposeness for what you want to do with it outside the context of those other regulatory decisions. This is something you might find out, well, wait a second, I really can't get this model to work. Maybe, then I have to take an alternative approach. So it's good to have this early discussion about setting that modeling tool in place. Then you can help, if you do that, then during the application review isn't going to be, spend multiple months on is your model okay? It's sort of like, well, we understand the model is appropriate. Now our reviewers are really looking at, well, what you propose to use it for and doing that evaluation. And that I think can bring things, that's much more efficient for us and, likely a faster evaluation for you. You'll get the comments and questions on your regulatory question earlier. Instead of having a mid-cycle with a lot of questions on your model verification, you'll have a mid-cycle meeting, on the fundamental questions of approvability first. And I think that's a big practical advantage of using this program to get that piece and get that piece in place and make the evaluation process faster to get to your regulatory decisions.
Oh, thank you so much, Dr. Leinberg. That's very insightful.
Since we lost Lucy.
Lucy, can I make one more point?
Yeah, please.
Can you hear me? Okay. So the other thing is, sort of expanding on what Rob said, and when it ultimately comes to the review team, I mean, you have to also understand how to use some of these meetings, this pilot and GDUFA meetings in conjunction to get the best input from the review team. So for example, you can use actually the pre-sub meeting if you're coming up with sort of a novel approach using a modeling and simulation approach, you can actually utilize that meeting to kind of inform the review team before you even submit and say, hey, here is our path. We already had the MIE meeting, and now we are following up. We put our data together, and this is what it looks like, and you will be and kind of walk the review team through and kind of utilize that meeting to your benefit. So I just wanted to plug in that information, which may be useful as an applicant. But again, it's sort of some of these meetings are available, and you may want to use it creatively to get the best input, before the actual review gets started even. So
yeah, Lucy, if I may, can I chime in a little? I really appreciate the talk on that. I think another element of the meeting is for non-complex products. For example, for the oncology products, which is non-complex, but we are currently recommending steady state PK crossover B study. We all know the study is costly and the patient recruitment is very challenging. And this MIE meeting can, model-based approach can potentially reduce the duration of study and the sample size. Just want to encourage industry colleagues to think from all around where the model can generate some value for the program. Thank you.
Thank you. I really want to take this time to thank you all for sharing your comments, your inputs, and those are all very helpful for our industry colleagues to have a deeper understanding of this new initiative. So, Dr. Rich, we actually got a couple questions from our audience. Do you mind stay on for a couple more minutes, moving into the next Q&A session?
Thank you for the wonderful panel discussion. We will transition to our Q&A panel, and our attendees may have noticed a slight shift in your screen as we transition to our Q&A session when the panelists disconnected their video, and then some were remaining online with muted mic for those attending our Q&A session. And if you haven't had a chance to submit your questions into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. We do have a few questions coming in right now. The first couple of questions we have are for Dr. Liang Zhao, and here is the first question. Can you share any experiences where an FDA pushed to go for the pre-ANDA meeting request rather than for the MIE meeting?
This very premature but a very critical question. So I think Altheria has covered the difference between pre-ANDA meeting and the MIE meeting. I would think that based on our observations, for example, for some orally inhaled drug product, we've been constantly seeing that the communication for modeling approach could be better held at the beginning. If we have that later, then that will lead to multiple cycles and potential more like a post-CRL scientific meeting. The scenario is that if the meeting have been, all the expectations have been clarified during the MIE pilot meeting in conjunction with the pre-ANDA meeting, then I think the applicant, the generic colleagues would enjoy a smoother ride down the road, even though you are spending a little bit more time at the beginning. Having said that, we really don't want to overburden our industry colleagues. We understand, the data package for the MIE meeting may not be fully there yet. So, but we do appreciate if you have a clearly thought modeling analysis plan and the data for model verification validation and the rationale behind that could benefit you a lot. And since we haven't conducted the pilot yet, so that's why I'm saying this a little bit of premature question. Certainly, we will share and improve the process along with the meeting experience. I presented in the slide, we'll have to evaluate the meeting process after 5 meetings conducted or after 1 year. So we'll keep everyone posted. We want to work together with all the stakeholders, including industry colleagues, to optimize the process. Thank you.
Thank you for responding to that question. We do have another question for Dr. Zhao, and here is the next question. Would the MIE industry meeting pilot be before or after the pre-ANDA meeting?
It can happen either way. Initially, the meeting was designed before the pre-ANDA meeting, but having said that, we realized that in the pre-ANDA meeting package, you could have an overall regulatory pathway. Then if we agree on the role of the MIE in the regulatory pathway, then the details could be followed from pre-ANDA meeting. Or you can also have a MIE pilot meeting before the pre-ANDA meeting. Then you can pose the question, can we use model for this regulatory role? And in the overall package, what are the, and these are the details, you can certainly do that as well. So we want to make it flexible at this point. Thank you.
Thank you for responding to that question. We do have a few more questions coming in. The next question will be addressed to Dr. Eleftheria Saclazao, and here is the first question. Is the model master file framework within the scope for the MIE pilot program?
Thank you very much for this question. Absolutely, the model master file framework can be discussed within the MIE pilot program. We encourage applicants to engage early in their development program to discuss relevant considerations that may apply to the development of a model master file, and especially if they intend to submit one or to reference one with their regulatory submission. And if you are interested in learning more about the Model Master File Framework, the Center for Research on Complex Generics and the FDA is co-hosting a public workshop on May 2nd and 3rd, and there will be additional information on that workshop released very, very soon. Thank you very much for the question.
Thank you for responding to that question. We do have one more question this round for Dr. Saclazao, and here is the question. What is the biggest benefit of MIE over a pre-ANDA meeting?
Thank you for this question. I believe that speakers as well as panelists have underlined that under the MIE pilot program, applicants have the opportunity to engage very early in their development program with the agency. So that is a great opportunity for them to get feedback very early. And when they're focusing on decisions on their drug development program. Of course, a complete package is necessary when they are submitting a meeting request. But as I noted in my presentation, a complete comparative modeling and simulation approach for both the reference and the listed drug and the test product may not be necessary. So the burden for data is significantly lower for this pilot program compared to the product development meeting. I think another important advantage of the MIE program over the pre-NDA is that the feedback you may receive could apply to multiple products you may have in the pipeline. So engaging early and receiving feedback on multiple products could be beneficial. And I'm not sure if Dr. Zhao also has feedback in response to this question. Thank you very much.
Yeah, my only addition is that, for the MIE pilot program, we do entertain complex modeling issues for non-complex products. So before that, I don't think we have a good defined venue for those type of discussions. Thank you.
Thank you both for responding to that question. Moving on to our next panelist, we've got a couple of questions that came in from Maria Monroy-Osorio. Here is the first question. Is it possible to request a meeting without having an ANDA number? For example, if a consultancy company or a CRO would like to have a meeting to discuss MIE issues that are not related to any single specific ANDA number.
Thank you so much for that question. As part of the pilot program, meeting requests are product specific, and therefore they do require a pre-assigned ANDA number. However, as Eleutheria was mentioning, as well as Dr. Zhao, if an applicant has one model that is being applied to multiple products, a meeting package can still be submitted for each of the individual products. What we request is that an individual package be submitted with the pre-assigned ANDA number, and then we will treat those meeting requests as a single meeting. And then in our final external meeting, the MIE issues discussed can potentially apply to those different products.
Thank you for responding to that question. We do have a couple more questions for Maria Monroy-Osorio, and here's the next question. What would be the average time for granting an MIE meeting for sponsors looking for guidance from the agency?
Thank you for that question. Once a meeting package is submitted and received by FDA, the grant deny decision will be made within approximately 14 days of the FDA receive date. If the meeting is then granted after those 14 days, the meeting will begin stage 2, which can be approximately 120 to 150 days, depending on the optional orientation meeting. And then we will conclude with stage 3, which is approximately 30 days. The total average time from the MIE pilot program can be approximately 154 days to 184 days, depending on the meeting being granted. And again, just to reiterate, the grant deny decision will be made within approximately 14 days of the FDA receive date. I do want to specify, and as a reminder, this pilot program meetings are not GDUFA-3 meetings, and therefore they are not subject to the performance goals. However, FDA does remain committed to the individual processes and timeframes that we have discussed today. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we have a couple of questions that came in for Dr. Andrew Babiskin. And here's the first question. In your experience, what MIE pivotal factors have consistently played a crucial role in gaining regulatory acceptance for computational models and the generic drug approval process?
Thank you for this question. It is a fairly general question because if you remember the content of my presentation, I presented on many different modeling disciplines that could be utilized for generic drug development. So I'm going to more take this question also from a general perspective as well. And what makes a case successful is really the ability to prove that you're able to properly input what the test and reference product is into whatever methodology you're using, but then also showing that your method is able to distinguish between any difference and able to include products that are not bioequivalent. You conclude that they're not bioequivalent. This is what we typically call type 1 error, that we're not having any false positives there. In this, I don't think this principle is any different than what you would even do for non-modeling, like in vitro-based methodologies. Like if you're developing a new analytical method, you want to show that as measuring what you expect it to measure, and you want to be able to show that it's able to discern any differences. That's the same expectations we would have in the modeling. So that's my very general response to that. That the critical part of your MIE approach is definitely having robust model verification validation that is towards the intended purpose. I believe I also have another colleague that also wants to respond to this question.
This is Fang Wu. I would like to add on some comment regarding using oral PBPK to conduct research risk assessment, a support waiver of in vivo BE studies. Important thing is incorporating appropriate in vitro testing results, for example, in vitro biopredictive dissolution as inputs to the model and conduct VBE simulations with good study design is also very important. Thank you.
Thank you both for responding to that question. And we happen to be moving on to Dr. Fang Wu, Senior Pharmacologist. And we do have a question that just came in for Dr. Fang Wu. And here is the question: how can industry use this MIE pilot program for oral non-complex products?
Thank you for that question. We know that scientific issues for non-complex products are currently not the main focus in the existing meetings, for example, pre-ANDA meetings. The requests focusing on non-complex products with complex approaches, like modeling approaches, are considered as a priority for MIE pilot program. The applicant can use the MIE pilot program to discuss modeling approaches in the development of oral non-complex products. For example, the applicant can discuss if and how to use the proposed oral PPK modeling for a scientific discussion for oral drug products. And the same applicant may also use this MIE pilot program to address common issues for multiple oral products, for example, conducting risk assessment, supporting waiver of certain in vivo BE studies for oral products. Thank you.
Thank you for responding to that question, Dr. Wu. We do have another question just came in about also oral non-complex products. And here's the question: what are the main topics of discussion in the MIE pilot program for oral non-complex products?
Thank you for the question. As mentioned in Dr. Babiskin's presentation, the topic for using the MIE pilot program for discussing the modeling approach for oral non-complex products include the risk assessment of deviation of dissolution profiles and the impact of particle size distribution on BE, and identify biopredictive dissolution, support BE evaluation, and also support BCS-based waiver and wave of fat BE studies for high-risk products, and wave of BE studies in subjects with gastric pH change, justify BE study design. In addition to that, the applicant can also discuss questions related to the feasibility of the oral PPK approach and the detailed information related to the model or platform development, verification, validation, and application strategies. Thank you.
Thank you for responding to those questions. Moving on to our next panelist, we do have a couple of questions that came in for Dr. Ming Hu, lead engineer. And here is the first question: how can machine learning help?
Hi, this is Meng Hu. Thanks for the question. I think here I don't need to speak for machine learning for their strengths and merit, letting alone some eye-catching, impressive application like AlphaGo, AlphaFold, even the ChatGPT. And also FDA, CDER already published guidance about how the AI machine learning can be used for the medical device submission application. And also, we have already seen a bunch of research has been reported, published, talking about how the machine learning can be used to facilitate the optimization of formulation and dissolution, even like in silico immunogenicity prediction. But in the same time, we acknowledge for the generic drug side, use of machine learning is still in the starting stage. However, we are so excited to see the forthcoming application using the component of a machine learning, AI machine learning models, and our internal AI experts and are more than happy to discuss with industry how the machine learning can be used to facilitate generic drug application and the regulatory assessment. Thank you.
Thank you, Dr. Hu, for responding to that question. We do have one more question for you that just came in, and here is the question. Providing a little bit of background, we need to apply logistic regression to support our bioequivalence analysis. If we'd like to discuss the use of logistic regression, which is not a typical machine learning algorithm, does it still fall in the scope of the MIE pilot program?
Yeah, again, thanks for the question. So the logistic regression, it's not, we agree it's not the typical machine learning approach, because usually machine learning approach will consist of empirical, data-driven, like inexplicit formulated algorithm. And if we remember the Andrew's presentation, we named some potential application under the MIE program, but it did not cover everything. So although logistic regression is not a typical machine learning approach, but it still belongs to data analytical approach. So if you want a focused, concentrated discussion on this perspective, which cannot be achieved through other meeting program like a pre-NDA or even control correspondence, I think MIE program is a good venue to go. And when you can submit your questions through the MIE meeting, then we can get started from there. Thank you.
Thank you for responding to that question. We'd like to bring in Dr. Bhagwat Reddy for a question that came in here during the Q&A panel. And here's the question for Dr. Reddy. Is the MIE initiative applicable only to ANDAs or also for the biopharmaceutics, PPI, particle size, scale up, food effect, formulation change, dissolution specs, product quality questions of an ANDA in the Division of Biopharmaceutics?
Yeah, so the MIE program is applicable only to ANDAs. For biopharmaceutics questions, modeling and simulation questions related to NDAs, please use Type C meetings that are already available. And that way we can get your questions answered there. Thank you.
Connect.
My apology. I think I was on mute. And let me read that question for Marie Monroy-Osorio. Here is the question. Can a decision to deny a meeting be appealed?
Thank you for that question. As part of the pilot program, meeting decisions cannot be appealed. However, if a meeting is denied, the applicant does have an option to pursue other communication pathways with FDA consistent with FDA's normal procedures. These communications may include controlled correspondences or pre-end meetings. We recommend that for additional information and when a pathway may be utilized can be found in FDA's guidance for industry, controlled correspondence related to generic drug development, which was issued in December of 2022 for controlled correspondences. And if you are looking for more information on pre-ANDA meetings, we refer you to FDA's guidance for industry, formal meetings between FDA and ANDA applicants of complex products under GDUFA, which was issued in October of 2022. Thank you.
Thank you for responding to that question. We do have a question that came in for Dr. Parther Roy from our panel discussion, and here is the question. It was mentioned that reviewers are present in MIE, but are they not also present in pre-ANDA meetings?
Thanks for the question. Yeah, the BE reviewers are present in both. So all of the pre-ANDA interactions that you that you see, including the MIE. What I think I was kind of, let me make it a little bit more clear. Sometimes you have these meetings, very early in the development, and then maybe many months and years have passed, after which the ANDA comes in-house. At that time, we try to assign the same review team within the Office of Bioequivalence. But as you can appreciate that since some time has passed, we may be dealing with some workload or resource issues. So we try our best to have the same review team, but there are times when we are not being able to. So we probably have another review team who kind of looks at it. But again, whoever it is, we just go back to the regulatory history, and we are very familiar with all of the interactions that happened with the company before. So nothing is lost, in that process. So, but I wanted to assure you that yes, the Office of Bioequivalence is very much involved in a lot of the interactions that you have in the pre-ANDA, and we have the knowledge and the information that is needed when the ANDA comes in house. Thank you.
Thank you for responding to that question. We do have a few more questions that came in for Dr. Liang Zhao, and here is the next question. Can I have an overall regulatory pathway question in the MIE meeting?
Yeah, absolutely. You can have overall regulatory pathway. Actually, it's indispensable when you discuss a modeling approach. Then the first question would be, what would be the role of the modeling in the full regulatory pathway? So therefore, absolutely you can pose a question on that. Can we use modeling approach in this case, for example, in lieu of conducting of a comparative clinical endpoint study? Can we then we know what would be the utility of the model. Thank you.
And thank you all the presenters and all the panelists for the great presentations and answering numerous questions that came in. We'd like to bring back Dr. Robert Lineberger for some closing remarks. Please join me to welcome Dr. Lineberger.
Thank you again, Roy. It's very great pleasure to be here today to listening to this meeting. And I hope that all of the attendees are looking for ways that this new pilot program fits into the development program. It won't be effective unless people make submissions to this, but we think that the time is right. We think that the use of modeling and simulation approaches will be an essential part of future product development programs. They're so useful for identifying what are my critical attributes and having discussion with the FDA. But also want to remind you as well that these modeling and simulation approaches are very useful for things that you don't need to talk to FDA about. As you're developing your formulation, as you are designing your pivotal BE studies, quantitative approaches can be really essential to getting that product right the first time. And you don't have to meet with FDA for things that affect your internal decision processes. So I want people to understand as well that the value of modeling and simulation, advanced quantitative methods, machine learning approaches can be used by generic drug developers right now to make their processes more efficient, increase your likelihood of successfully passing your BE studies. And we encourage you to build on that use as you find these things useful to the meetings with FDA, or when I want to bring these things into the discussion with FDA. So like we're going, you either foresee or you get questions that FDA is going to ask you for, well, you're doing something different. Our favorite reviewer thing is, can you provide a justification? I think the modeling and simulation is an important part of that justification to say, especially if you've used them in pharmaceutical development, then you have confidence that they help you make good decisions, and you can bring that in as part of the justification for answering the regulatory questions. And so for cases where you're going to bring the model into the application, not just using it for your own internal decision processes, I think this pilot program can be really effective in getting focused feedback on the modeling questions. So I think one of the reasons, and the question that came up in this was, well, why should I use this meeting versus, a normal product development or pre-ANDA meeting? It's because you want to because there's a need to focus time directly and focused on the modeling conclusion here. And this can be more efficient for FDA. We can have a leaner team. They're focused on that question. You can get a result driven on that question, much more appropriately, rather than having multiple cycles of product development meeting. If the first meeting says, well, look, here's the ways you need to improve your model, then come back again to ask your regulatory question. Here you can get the modeling part done, and then there's a separate discussion around the regulatory question that has maybe a bigger group of participants and a broader aspect of it. Or you might use this meeting after. You might say, well, look, we really need to, FDA might tell you at a product development meeting, we really need to see your modeling approach in the application to decide if what you're doing is appropriate. You might want to try to have one of these meetings to really work through the details of the verification program. So when you submit it, that's not going to delay your review process. So there's a lot of ways you can strategically think of how to use this meeting, but it's really an opportunity to get detailed and focused feedback on the modeling approaches. And then use that to streamline the other regulatory interactions you have with FDA, because your model is already being, my model is already validated. You're not sort of arguing, is the model correct? You're talking about in other interactions, the regulatory use of the model, because you've had this meeting and you've come to a sort of agreement on this is the data that supports the model's fit for the purpose that I'm using it. And so it really is a foundational piece to making smoother, faster, more precise regulatory interactions through other meetings and through the ANDA review process. And so we hope this will be very successful. It's a pilot program, so we want to, and we're not going to learn from this pilot program unless people make submissions to the program. So we really encourage you to look at your ongoing development programs and say, are there places where modeling is going to be important to my ANDA submission, and use it and use this meeting to get feedback on those models. This will really help us improve the meeting process and find ways to make this model-informed meeting, going from a pilot to something that's continues. We really need participation in the pilot program to get that feedback, to really understand with practical examples how useful this is. So, the best thing you can do to make this successful is submit your meeting packages to this program so we can, use it as part of the pilot and really develop it. With that, I think I'll pass it back to Ray for some final remarks. But again, I want to thank all of our SBIA staff who've done the logistics, and our speakers from ORS and other offices, and panelists who have worked on this webinar, and we hope to interact with you through this meeting process. So I'll turn back to Ray. Thank you all very much.
Thank you, Dr. Leinberger. And we do have a few housekeeping reminders for everyone. Please remember the 14-day deadline, and that's February the 1st, to claim the Certificate of Attendance to support CEs for SOCA, RAPS, SQA, and ACRP. Remember to download the certificate immediately upon creating the certificate. Otherwise, this opportunity will be lost. Recordings will be posted in approximately 7 to 10 business days. On behalf of CDER Small Business and Industry Assistance, we hope that you enjoyed today's presentations and found them helpful. We look forward to your presence at our next SBIA event. Thank you for attending, and this concludes today's virtual event.